This is a scanning electron micrograph of a biofilm, and this thing's probably about 30 microns across right here. Maybe this is 15 microns across. And there's a series of diatoms that are in the biofilm. We've already seen, I suspect this one's Melisira and maybe Navicula or Gyrosigma, I can't quite tell. But these are all the silicon, and then this mucopolysaccharide matrix that they're inside of. And the idea of this is that when we have microbes together, they're actually all interacting with each other. They're in close proximity with each other. And most of those interactions are going to be mediated by chemicals. And then there's going to be bigger things that eat them. Okay, so we're starting now with the organism interaction stuff. We're going out of the nutrients and into how organisms interact with each other. We'll go through a series of them and build the ecosystem up to the whole ecosystem. So first I'll talk about microbial behavior, how the different interaction types that occur, and then go through the series of interactions that might occur. So one of the most basic things that microorganisms can do is they can either sit still or move. And if they move, they need to have a reason to move, right? That evolution suggests that if you just move for no reason, then you're wasting energy and you're not going to be able to compete against the one that sits still. So if you're going to move, you have to be able to move towards or away from things. You have to either get away from bad stuff or towards good stuff. Right? And so organisms can do this through phototaxis. They can be attracted towards light, or negative phototaxis would be attracted against. So phototaxis, right, towards light. Geotaxis, they can go after gravity, and we'll get to this in a second. It's pretty cool. They can follow chemicals, chemotaxis. Magnetotaxis turns out to be another form of geotaxis for the most part, magnetic fields. So we'll start with magnetotaxis. There are these bacteria that this fellow Blakemore was looking at under the microscope, and he saw they were all swimming one way. He had this culture of marine bacteria. He saw they were all swimming one way. And for some reason, he moved the magnetic field in a flip direction, and he couldn't figure out why this was. And he looked at them on an electron micrograph, and you see these iron particles, magnetite in there. And found out these bacteria had little iron particles that they were synthesizing and placing inside of their cells. And the reason they were doing this was to be able to move down into sediments. There were bacteria that were isolated from sediments. And the reason that they can move down into sediments if they follow a magnetic field is that the Earth's magnetic lines, if you have the globe here, right, the magnetic lines in the North Pole are kind of going down into, they're not straight north-south. Like if I have a compass right now, and it's facing north, it does face north, but the needle also has a little inclination downwards because of the way the magnetic fields are shaped. So if the bacteria can follow that north, it'll eventually get itself down into the sediment. So it doesn't have to have any light, it can just go down with that. The cool thing is that when they isolate the same species of bacteria from the southern hemisphere, they go to the south because the south facing is down in the southern hemisphere. So these occur in marine and fresh waters now that they know. So we can talk about the other ones, you know, are a little bit less gee whiz. I mean, they just have an eye spot or something and they can see light and integrate over time, or some sort of chemical sensor like glucose that says, okay, concentration's increasing, I'll keep going this way. And we'll talk about how they do that. The next issue, though, is how fast things move. So we can think of absolute velocity in terms of meters per second, or relative velocity in body lengths per second. And we have a series of things we can, you know, salmon or trout can go pretty good. They can go 44 to 6 meters per second, and maybe 8 to 10 body lengths per second. and that's better than some of the slower swimming fishes. But it turns out a ciliate protozoa can swim about as fast as a salmon relative to its size. Right? It's going about 10 body lengths per second. Copepods are able to kind of jump and go really fast for short periods of time. They're actually the all-time relative fastest swimmers at 200 body lengths per second. So copepods are pretty cool. But even things like bacteria and diatoms can glide on solid surfaces. They can move along, at a fair pace, not huge pace relative to their body size. But since they're microns across, though, this is, we're talking tens of microns per second. The random model is one of the simplest ways, probably not the simplest way to have a taxis, but it's one of the simplest ways. The simplest way is if things are bad, move, and if things are good, stop. But this is a very commonly used method, including salmon trying to find their natal stream, bacteria trying to find a source of nutrients, and many different organisms use this type of approach to finding source. So this would be some source of attractant of some sort in both still water and then perhaps with directional flow. But this could work with an airborne particle too. But for aquatics we have this. And the rule essentially is that if things aren't getting better or getting worse, you stop and change direction. So you go for a while, and if things aren't getting better or actually getting worse, then you stop and change direction. But you change direction only in a random sense, randomly, right? So because you don't have any information as to what the true answer is. So you can see this thing is just sort of randomly wandering around, and then eventually it hits the edge of the concentration gradient, and it keeps going until as concentration increases, and then concentration starts to decrease. But it doesn't know what direction after that it's supposed to be going. So it just randomly picks a new direction, goes, nope, getting worse still. Randomly picks a direction, nope, still didn't do anything. Randomly picks a direction, yeah, now it's getting better. And eventually you'll see this can concentrate organisms to where they want to go. And it works whether you're talking about a directional plume or just a point source. This essentially just requires organisms to be able to sense what it is they're trying to get towards, number one, and two, integrate the changes in the concentration of that over time. So it's a very simple set of rules, but it's a very common way that people even search that way sometimes, but many organisms do. Okay, so now movement, we've talked about movement, and then we're going to talk about some of the different ways that organisms interact. And some of these are really general, and we'll come up with the way all species interact, because they're just other species. And the first idea is the idea of density-mediated versus trait-mediated interactions. And so density-mediated interactions is essentially an interaction with another organism that depends upon your density relative to their density. So this might be, for example, competition for food. If you eat a bunch of food and the other one can't get it, then you're competing with them directly, right? This is density-mediated. Or predator-prey interactions that are dependent upon the density of the predator and the density of the prey. Trait-mediated interactions are less direct in the sense that there are ways that species interact that have arisen from evolution that oftentimes essentially keep them from interacting in a density-mediated way. So this would be, an example of this would be, say, a predator avoidance response that's evolved, where it doesn't matter how much predators there, you just don't go where predators tend to be, right? Or a competitive specialization, niche specialization, where you've essentially evolved away from competing with another organism by specializing on one part and the other organism specialize on the other. So we've talked about high light versus low light species, or high nutrient versus low nutrient species. All of those would be trait-mediated interactions. And then the other thing we need to think about is that organisms have direct and indirect interactions. So really, the vast majority of species don't interact directly with each other. There are, for example, bacteria floating all over in this room, right? And we don't have much of any interaction with those bacteria at all. And we don't affect them very much, they don't affect us. Right? But indirectly, maybe we do, because we'll do things like, I don't know, change the cockroach population in the room that perhaps would be eating them or living on their frass or something like that. So we have a lot of indirect interactions where you affect or interact with organisms, and a lot of direct interactions where you don't. And it turns out that if you think about any two species randomly grabbed from the environment, that they will tend not to interact with, be interacting. The chances are good that they won't interact with each other. So we need to be thinking about these types of interactions as we go through this and essentially the next 3 or 4 chapters that we talk about. So in microbes, we first talk about predation and parasitism. In the first part of that, we'll be talking about viruses. Then we'll talk about how animals and protozoa can eat small cells. We'll talk about functional feeding groups on whole assemblages of microbes and selectivity of particle feeders. The way microbes adapt to avoid predation, and finally talk about parasitism and other exploitative interactions. And so if you remember back to my initial definitions of the way species interacts, predation is not the only plus-minus interaction. There are other ways that organisms can exploit organisms and have negative effect on them and not be predators on them. So one of the first things to talk about, let's see how this is. One of the first ways to talk about is why microbial interactions are important to aquatic food webs. And this is an evolving view of the world in aquatic food webs that has changed. Probably the revolution in this change in understanding in general occurred about 20 or 30 years ago. And so the traditional view of aquatic food webs were that you have these nutrients, and these nutrients were able to stimulate growth of the phytoplankton, and what was really important was the stuff you could catch in a plankton net, because that's the technology that people had. That zooplankton ate that. Perhaps there's carnivorous zooplankton that would eat that. And we have planktivorous fish that eat those, and then maybe 1 or 2 levels of predators on top of that. And that when those organisms then excreted, they put nutrients back into the system and sort of complete that pathway. So this is a traditional view, and the more modern view is the idea of a microbial loop. And the idea that there's this very rapid cycling of materials that's going on within the microbial community, and then the animals are sort of skimming off the top of that they're not sort of in the central line of things. And so we have nutrients and organic carbon. And there's a lot of dissolved organic carbon around, right? And if that isn't used by the food web, it's lost in the system. So the bacteria, heterotrophic bacteria, can use up that organic carbon. These have got a bunch of liquid nitrogen coming through. And the other thing that happened was as people got the ability to filter out very fine particles on membrane-sized filters with controlled pore sizes, they found that there were photosynthetic organisms that were the size of bacteria. Close the door there. Usually it's quieter around here. So they found that there are photosynthetic bacteria that were caught on, say, 0.2 micron filters, or bacteria-sized organisms. And when they found this, they realized that there was a whole group of primary producers that was out there that was ignored when net phytoplankton. And these can occur in lakes, and particular marine systems, and they perhaps are responsible for 1/5 of the primary production on Earth, actually. So it's a big missing hole that people figured out about. Well, if all these things are really small like this, bacteria and fecal phytoplankton, it's not necessarily clear that the zooplankton, we'll talk about why, the zooplankton are real efficient at eating those things. They can eat them at low rates, but the ones that turn out to be really the most important are protozoa. And then rotifers also can eat those and can eat the protozoa. And rotifers can graze on these different things. And we've seen rotifers and protozoa in the laboratory. They're common, they're abundant in many freshwater systems, and this is how they fit in. And then eventually these carnivorous zooplankton can perhaps eat these protozoa and the rotifers as well as this phytoplankton. So we have essentially this microbial loop going here. Then we have all these organisms excreting out nutrients and organic carbon and feeding this microbial loop. Okay, so it's a different view of the things, and microbes are way more important than people thought traditionally, and that's because the methodology evolved so that we could really get at sensing how important they were. So one of the things I've talked about is all this organic carbon that's leaking out of everything and the nutrients that leak out of everything. We sort of have talked about this when we talked about heterotrophic activity and stoichiometry and nutrient mineralization. And one of the big reasons for this is all organisms have viruses. So these bacteria, these protozoa, everything is essentially subject to being attacked by these viruses. And it appears that they can control algal blooms and bacterial populations at times. So the research that's gone on has shown that organisms like cyanobacteria that form very dense blooms and unispecific blooms actually can be resistant to viral attack, viral infection. And they have to evolve that ability essentially, and it somehow costs them energy. Because if you take them, we got a fly going in here.
It's a bee.
Huh?
It's a bee.
It's a bee. It's a bee. If you take where was it? Oh yeah, so if you take these species and put them under low virus loads, they're unable to compete with the species that are not resistant to the viruses. And so we have organisms that are basically trying to keep off these viruses. And you can imagine that just like when humans occur in more dense populations, that the epidemiology is such that diseases can pass more readily among them, right? It's the same is true with microbes, that if you have a dense microbial population of one species, that's where it's most likely for the viruses to pass through them. And the reason for that is that these are inactivated in environment, in natural environment by sunlight, and they can get stuck on particles. So you need to have essentially another host for the virus close by so it doesn't get inactivated before it gets to you. There's been a lot of research on inactivation of environments, viruses environments, mainly related to human viruses. So human diseases are also caused by viruses, and how long they last in, say, groundwaters or streams or rivers is of important concern to public health experts. But this is also true for the viruses that influence all other organisms that are out there. Fish aquaculture people have problems with this. There's a number of viruses that infect fishes, and they'll have to destroy all the fish they have so they don't spread it, and there's not necessarily treatments for it. So one of the questions is, well, if all these viruses out, these really small particles, why isn't the entire world's carbon tied up in viruses that are floating around? And the reason is that there are actually organisms that can eat and engulf these very small particles, the protozoa. They actually can eat a particle that is perhaps 1/10 of a micron across, around in that size. So remember, bacteria are about a micron across, and viruses are 1/10 that size or smaller. Most are. There's some giant algal viruses that are almost the size of bacteria, but that's another story. And one things that's really important about this is that viruses can move genetic material. Because what they're doing is they're taking advantage of the host's genetic material to reproduce. And they sometimes include other genes inside of the virus and then go out and infect another organism. And if it's a defective infection in the sense that they can't reproduce themselves, then that DNA gets dumped into the other organism and it moves into it. So it's actually seen DNA move across taxonomic lines this way. And it's actually a method that molecular biologists use to genetically modify organisms. They'll have a defective virus that inserts DNA in but doesn't continue to reproduce. So it's a natural process as well. But it means it increases the diversity of microbial populations and even the microbial populations that don't have sexual reproduction to directly exchange genetic material. Okay. So they can exchange plasmids, but it's another way that genetic material can move around the environment, and it's not necessarily strictly set into taxonomic lines. So they have ecological importance and evolutionary importance as well. So this was not really well appreciated until there was a colleague of mine that was interested in finding out with electron microscope micrographs how good that these filters are that have pore sizes. They're like they have nucleopore filters, and they use radiation tracks to make different size holes. And you can buy them, 0.2 microns, 0.4 microns, whatever. And he wanted to find out how good they were, so he put them on an electron microscope and found out they're not great because you can get a couple holes next to each other, and they're much more variable than the pictures that the companies put out. But he was filtering this lake water, and then he started seeing these virus-like particles on there, and found that they're extraordinarily abundant in the lakes, and they happen to be in Canadian lakes that he was looking at, and published on this. And people started realizing there's all these particles floating around in water that look like viruses, and they didn't know what they did. And then they'd find things like this, where they actually have a bacterium, and you can see these little blebs on the outside. There's viruses attached all over the outside of this bacterium, and it's one from a plankton in a lake. And then they started doing experiments like concentrating water that had very fine particles in it, say everything less than a micron, so too small for bacteria, but bigger than, say, 1/10 of a micron. Concentrating those particles and then putting them in algal cultures from lakes and watching the algal populations drop immediately because some of those were infected particles. Able to trace down the idea that they really were active viruses in the environment that were controlling populations, microbial populations in the natural world. So now, logically, it makes perfect sense that that's going on, but when I was a student at your age, that was not very well understood. So it's something that's changed pretty drastically since that time. Another part of the microbial loop, if you will. So I alluded to the idea that consumption of particles is important, and we are in a position now where we're going back and starting to talk about Reynolds number again. I promised you it would come up again, and here it is, rearing its ugly head. Okay. So if it is very small, if a particle is very small, the solution is very viscous, right? Remember, viscosity increases as you get smaller and smaller. So that means the traditional way that you might think about taking a particle out of solution, filtration, is worse and worse at getting particles out, and takes more and more energy the smaller those particles get. And if you've ever actually done filtration, you might know that if you use a very fine pore filter, it's very difficult to pour water through that, and that's because you're basically trying to pour a viscous solution. It'd be like using a coarse filter and trying to filter corn syrup through it. It just doesn't want to get through those spaces. So let's say you have a filter, like you're a Daphnia, and you have a filter that's a comb-shaped thing. The finer you get those, the more energy it takes to get the fluid through that comb, right? And eventually what's going to happen is the water will just go around it rather than it won't have enough force to get between those cones. So you can't really use a filter to get these very small particles out. It becomes physically very difficult. So at the lower end, you have this collection apparatus. You can't get the collection apparatus small enough to work with. And then at the upper end, you can't fit the stuff in your mouth. So we've already probably seen this. A number of you have had fish ecology. You know that there's gauge limitation. There's a saying with people, don't eat anything bigger than your head, right? It's the same thing. You can only fit so much in your mouth. In addition to this particle size, there is a functional relationship between particle concentration and the rate that things can be eaten. And in general, at the low end, you have to eat more to make a living, but at the high end, you can only eat so much, right? So you don't eat anymore. You can't eat anymore. And a function of particle quality. The organisms, they'll have to eat more if it's a low quality food, or they'll pick and choose and eat the best quality food that they can get. And overall, it turns out the greatest rates of microbial ingestion occur when the entire assemblage is consumed. So there's a lot of organisms out there that consume microbes and do so based on just eating a whole bunch of stuff. And that would include things like snails and mayflies that graze on rocks in the streams. The snails just have a radula and they're just scraping everything off, all the microbes in it, and eating it. Or a shredder, an organism that eats leaves. They're just chewing up the leaf, but what they're really eating is all the fungi and the bacteria, the microscopic organisms that are living inside of that. So if you can find a food source that has a big high microbial population in it and eat the whole thing, then that's one good way to make a living. You can think of the ruminants, they've actually sort of internalized that and they put the microbial population inside of them, eat the grass, and then they get that works for them. So some real data on size, and this is a bunch of different protozoa, ciliated protozoan species, and particle diameter is on the x-axis, and on the y-axis is the clearance rate, and that's how much volume they can put through. And this clearance rate then peaks out and then decreases for a number of these species. And you can see they're specializing on particular particle sizes. So some of them tend to specialize in the very smallest particles, and some of them on the very largest particles. And then, as you can imagine, if you specialize on smaller particles, perhaps it takes you more energy, and so you can't maintain super high clearance rates, but you have a more abundant source of food. So we've seen these trade-offs, right? We've seen trade-offs between low light and high light species. You can either photosynthesize well at low light or photosynthesis at higher rates at high light. Low nutrient or high nutrient species. You specialize on low nutrients, but your growth rate is going to be lower than if you're specializing on high nutrients. But if you're in a low nutrient environment, it helps you. Small particles or large particles. It takes more energy to use small particles. You can't grow as fast, but if you're getting into an area where you're competing with these guys for larger particles, and they can grow and clear particles out much faster and take them out of solution, you're never going to win. Right? So this is basically evolution of niches, if you want to think of it that way, and specialization on particular sizes of food. So you can think about this in terms of energetic constraints and in terms of particle concentration. And we have protozoa here eating bacteria, and we see that the higher their concentration is, the more they eat. But then at some point they're leveling off here because they can only eat so much, and then they can't grow any faster, and that's the maximum rate. Okay, we can see a similar thing with Daphnia. And this is a common one that I ask people to reproduce on tests because it puts a number of these ideas together. And so we have on this graph on the x-axis, we have the number of algal cells per cubic centimeters. And we have the filtering rate per animal here. And we see that if there's very low concentrations of algae, they've got to filter a lot of water, right, to get the food. And at some point, this is going to actually stop completely because they're just, they can't, it's such a low concentration, they give up. But you fit this point here, and then they get up to a point where they're super concentrated and they're barely filtering. They're just like, you know, every once in a while eating something because it's just right there. They don't have to move a bunch of water. And then their ingestion rate, though, the rate that they can actually get food in when a small particles, when this low concentration of particles is low, and then it levels off here. So these are inverse of each other. This is substantial. We can talk about, when we talk about what these ingestion rates are and these filtering rates, they're extraordinarily high. If you have a pond full of Daphnia, it can actually turn over the entire, filter the entire water several times each day. So they're working real hard to get these microbial populations out. And they're competing with each other to do so. So take home points. You ingest more and more at higher concentrations, but it levels off because you can only eat so much. But to ingest stuff at low concentrations, it takes a lot of energy. You've got to filter a lot of water, and at high concentrations, you're doing better. It makes sense. So I mentioned this idea of organisms that eat whole assemblages of bacteria. So the ciliates and the microflagellates are really eating one bacterium at a time. Ciliates might be able to take in lumps. And we have the rate of bacteria, the number of bacteria ingested per time, and this is a log scale. And we see that these things, these small microbial things, don't have the potential rates that some of these bigger organisms do, because the bigger organisms can take in big amounts. They can eat a whole leaf particle that has bacteria on it or something like that. But you can also see the ranges are extraordinarily variable as well. If you're a nematode, you obviously have to take in a lot more to survive than you do if you're a ciliate, just because of the way this is, the number ingested per animal per hour, right? If you have a big body, you need to feed it. I mean, these things are 100,000 times bigger than microflagella. So that's the other part of the equation. And I'm talking about this filter feeding. This is a rotifer. If you're an alga, this would be like a Halloween horror picture, right? This rotifer is filtering stuff, and it's basically causing the water to rotate, and then when a good particle comes in, it takes it. When the bad particle comes in, it spits it out. So it's continuously assessing the quality of the food that it has. And even the simplest organisms do this continual assessment of the quality of the food. So we talked about microflagellates, very small protozoa that are eating bacteria. If you do an experiment, then you can put in, you can label things two ways, and this is how they get these feeding rates. One thing they'll do is they'll put a radioactive tag on the outside of actual living bacteria. So it's just like the food they eat. They can measure how quickly the radioactivity gets incorporated into that animal. The other thing they can do is they can use fluorescent beads that are the same size as the bacteria. And so they can use a fluorescent microscope to see how many beads they get in their gut over unit time. And even those individual cells that, you know, the things that are as big as bacteria, the rotifers and the Daphnia are preferentially going after the food that's good, and they're rejecting more of those fluorescent particles. They're just plastic beads, basically.